Welcome back to part 5 of amino acids and nitrogen metabolism. The focus for this installment is the urea cycle. The metabolism of nitrogen-containing compounds generate ammonia, which is in equilibrium with its aqueous ion, ammonium. Some of the ammonia generated in the liver or transported here is recycled into amino acids, nucleotides, and so on, and the excess is eventually excreted as urea. We will see how this is done in the urea cycle. But first, we will look at ammonia production and the most important enzymes involved. Ammonia is potentially toxic, and humans produce about 1,000 millimole or 17 grams of ammonia daily, which is reutilized in biosynthesis or handled as waste. Excess ammonia produced in other tissues are transported to the liver, where much of this metabolism takes place, including the production of urea as a non-toxic final state of nitrogen. Now, high levels of ammonia, state called hyperammonemia in the circulating blood, causes many problems, with symptoms summarized in the anatomical figure here. Notably, ammonia that gets dissolved in the blood at higher concentrations, it could cross the blood-brain barrier quite easily, and this may trigger seizures, a coma, or in the worst case, death. Now, hyperammonemia may be caused by a defective liver, but it can also be caused by defective enzymes. For what else is handling this potential hazard? Yeah, that's enzymes. There are several enzymes tasked with removing ammonia in the form of ammonium ions. One enzyme is glutamine synthase, which is active in the brain, in the liver, and the kidneys, as indicated with the blue and green lines on this figure. So this enzyme produces glutamine from glutamate in a two-step process, converting the ammonia hazard into a non-toxic and useful and transportable substance. When imported into liver mitochondria, another enzyme called glutaminase can catalyze the release of ammonium again, and here it gets recycled or converted into urea. The normal concentration in the portal vein, that is the vein that leads into the liver, in the blood there, the normal concentration is about 300 to 600 micromolar of ammonia or ammonium ions. And after the liver has done its business, about 20 to 60 micromolar remains in the blood that leaves the liver. Okay. So glutamate synthase activity in the brain and kidneys constitute one ammonium handling and transportation mechanism. Another mechanism, another ammonia handling mechanism in humans takes place in the muscles. We've already mentioned this in an earlier installment of this lecture, but we repeat it here. So here, alanine, aminotransferases transfer amino groups to pyruvate, forming alanine, which is easily transported to the liver by blood. And this is another way of converting ammonium to transportable, non-toxic, useful amino acids. But wait one moment, this arm here, what if it was much beefier? I think we have to explore that a little bit. Aha! So in Trogdor, we have argued that ammonia production ends up in a gland near his gaping maw, where it is released together with other flammable products to produce his fiery breath here. This would explain the beefy arm, which not only is gloriously beefy here, but probably also a powerhouse of ammonia production. Now, as we have seen earlier, hyperammonemia can lead to death in humans. In Trogdor, hyperammonemia It likely contributes to his fiery breath. He is constantly charged with excess ammonia, and he lets it out in his fiery breath. Now, it could be argued that getting burninated is just another death by ammonia. However, we must also admit that burnination is by far a more glorious death. Now, there is actually another twist to this. Remember the symptoms of hyperammonemia? When hyperammonemia acts on the brain, it lists a couple of symptoms here, among them combativeness. And now Trogdor is definitely combative and aggressive and belligerent and warlike. It all fits very nicely, I think. So Trogdor's physiology is tailored around the perpetual state of hyperammonemia, which allows him to burninate the countryside and consume peasants at a very high rate in a vicious but also glorious cycle of destruction. So maybe in Trogdor, but definitely in other animals. The liver is a very important organ for handling ammonia in various forms. Let's look at nitrogen import in the liver and the processes that follow. Glutamate, glutamine, alanine, and other amino acids enter the liver, usually through blood in the portal vein. The amination occurs for all amino acids, but these are of particular importance, and we will therefore focus on these. Amino groups are removed from glutamate in the mitochondria by oxidative deamination catalyzed by glutamate dehydrogenase. This enzyme is a bit unusual in that it can use both NAD and NADP as a proton acceptor. This protein has 6 identical subunits and is allosterically regulated. Likewise, glutamine that enters the liver can be converted to glutamate along this pathway and shed an ammonium by the action of glutaminase. So glutaminase would then produce ammonium and glutamate, and then this enzyme can act on this glutamate too. Now, note also that aspartate can be produced by accepting the amine of the glutamate here in a transamination reaction. Remember those from part 1? The aspartate will enter the urea cycle here, and we will see it again later. So this pathway slots into the urea cycle, and we'll get back to it later. So just to look at this, it's the oxaloacetate that is subject to a transamination reaction where glutamate is the donor, and this produces the familiar alpha-ketoglutarate. The next step in ammonium processing is the production of carbamoyl phosphate. This involves ATP, bicarbonate from the solvent, and the ammonium ion. So the ammonium ion is produced here, bicarbonate from the solution, and ATP. So this process is of course catalyzed by an enzyme called carbamoyl phosphate synthase 1. And here is a schematic of its action. In the first step, a phosphate group from ATP is transferred onto the bicarbonate here. This creates a reactive anhydride. This is readily aminated, and the phosphate group is displaced. This creates carbamate. Carbamate is again phosphorylated by ATP to produce carbamoyl phosphate. So again, we see that phosphorylation creates a reactive substance that can be subject to more reactions, in this case, amination. Now it's this carbamoyl phosphate that enters the urea cycle, and that is a subject of the next slide. So here we're now focusing on carbamoyl phosphate, and how it enters the urea acid cycle. Note that the urea cycle spans the mitochondrial matrix and the cytosol of the liver cell. So part of it is inside the mitochondria of the liver, and part of it is in the cytosol of the liver cell. Now, as carbamoyl phosphate enters the urea cycle, we can identify 5 steps. 1, 2, 3, 4, 5. So 2 of those are step 2A and 2B. So the last one is actually labeled 4, but we'll figure it out. Now, in step 1, the enzyme ornithine transcarbamylase attaches it to ornithine that comes in here, resulting in a substance called citrulline. You can see the enzymatic pathway associated with this reaction in the panel to the left here. In step 2A, an intermediate is created by attaching AMP to the carbonyl oxygen of citrulline. This happens here. So this we have seen before. Again, it's a phosphorylation event that creates a reactive intermediate. Now in step, and here is the reactive intermediate. What makes this a bit different is that it's not just a single phosphate, it's the entire AMP moiety clinging on here. So this intermediate in step 2B is subject to aspartate that comes in from the transamination that happened in the mitochondria. Remember that we would return to this, and here it is, coming in like this. And this aspartate displays the AMP, and the resulting substance is called argininosuccinate here. Argininosuccinate is processed by argininosuccinate ASA in step 3 here. This produces fumarate, which later may enter the citric acid cycle as malate, and arginine, which stays in the urea cycle. You will remember this material from the biosynthesis of arginine, which gave us a head start on the urea cycle. So what we're going through now is essentially the same, only this time we have gone through it in more detail and with the emphasis on the whole urea cycle. So now we have arginine, a molecule with no fewer than 4 amino groups. In the last step here, arginase cleaves off 2 of these amino groups in the form of urea. Then what remains is ornithine. So we are back at step 1, and ornithine enters the mitochondrial matrix and gets ready to be treated with ornithine transcarbamylase. That is, getting a carbamoyl phosphate added to it. And this completes the urea cycle. Now, what about the urea produced? Well, it enters the bloodstream and is carried to the kidneys, where the blood is filtered and urea and other waste products are gathered and excreted as urine. And now, commentary on how different animals pee. So many terrestrial creatures, such as ourselves, convert nitrogen into urea and excrete it in urine. However, bony fish, like Nemo here, and the larvae of amphibia excrete ammonia directly, so they don't bother to convert it to anything. They can do this because the water they swim in quickly carries the ammonia away, so they avoid high concentration and therefore its toxic effects. Now, curiously, cartilage fish, such as shark, does bother to convert nitrogen to ammonia. Finally, we have Trogdor, the burninator, along with other reptiles and birds. They excrete nitrogen as uric acid, not as urea. So here is uric acid. Looks like a perine, really. Now, in the case of Trogdor, we expect that he does not really waste nitrogen in this way, but rather spends it as ammonia for his glorious fiery burnination. Okay, moving on. Now we will consider how the urea cycle and the citric acid cycle is connected through the aspartate argininosuccinate shunt. You'll remember it from the schematic figure I have started the lecture parts with. In the top left corner here, I've shown it again. So that it exists shouldn't come as a surprise. Here it is, the aspartate argininosuccinate shunt of the citric acid cycle, spanning the urea cycle and the citric acid cycle. The shunt shares malate and oxaloacetate with the citric acid cycle and argininosuccinate with the urea cycle. Argininosuccinate is processed to arginine going this way, and fumarate and oxaloacetate going this way. Now, fumarate and oxaloacetate is processed to malate in the shunt, and malate enters the citric acid cycle here. Notice the malate aspartate shuttle, which connects aspartate and malate. So, but now the shunt shares malate and oxaloacetate with the citric acid cycle and argininosuccinate with the urea cycle. Argininosuccinate is processed to arginine and fumarate. Arginine going this way and fumarate going this way. Fumarate and oxaloacetate is processed into malate in the shunt, and malate enters the citric acid cycle. Notice the malate aspartate shuttle, which connects aspartate and malate. Now, by now, we have actually gone through practically all processes in these cycles. The main takeaway message from this slide is that these cycles are connected, and that argininosuccinate here, malate, and oxaloacetate here and here constitute the compounds that they share through the aspartate arginine succinate shunt. It is natural and very important that they are connected, otherwise glucogenic metabolism of amino acids will not be able to handle the accumulated ammonia. Instead, it can turn it into oxaloacetate and enter the urea cycle as argininosuccinate. At this point, you might ask what determines the pathway of oxaloacetate. So what actually determines whether a pathway will go this way or this way? Why does it sometimes go into the urea cycle to donate nitrogen and sometimes not? Well, this depends on the metabolic state of the organism, and it is handled by regulation. The regulation is met in the long term by expression of enzymes involved in the urea cycle, and its entry point in the mitochondria. That is the enzyme carbamoyl phosphate synthase 1. All of these enzymes are expressed at high levels in starving animals, which will use muscle proteins as fuel in the citric acid cycle, causing much excess nitrogen to be shunted into the urea cycle. The same pattern is seen in animals fed on high protein diets. Conversely, protein-free diets lower the levels of these enzymes. The second level of regulation is much quicker in responding to circumstances and involves allosteric regulation of carbamoyl phosphate synthase. This is depicted here. This entry point is activated by N-acetylglutamate, here, a product of acetyl coenzyme A and glutamate. The enzyme responsible for creating this activator senses and is in turn activated by the presence of arginine. So N-acetylglutamate synthase producing this activator is itself activated by arginine. So remember, arginine has no less than 4 amine groups, and as such is a good indicator of the amount of nitrogen available in the amino acid pool. So the brain is full now, presumably. This is the last slide of part 5 and the last slide of this entire lecture. Thank you for watching.